Okay, so this is the last part of the class. After B cells complete their development and they get activated in the secondary lymphoid tissues, they start producing antibodies. How do these antibodies protect us against different pathogens? Here I'm going to use some clinical examples in order to explain the function of antibodies or the ways that antibodies protect us against pathogens. And I know that you guys have not yet started your clinical training, so that's fine. We are not going to go to too much detail about the clinical manifestations of these diseases. We're simply using them as examples to learn about B cell and antibody function a bit better. And the first picture that I'm using is from CDC website from a patient with tetanus. And you know that tetanus is an infectious disease which is caused by Clostridium tetani. And the way that it works is that it produces a toxin. So somebody gets infected, somebody gets a wound which is contaminated by maybe soil or some other things which have the spores for the bacterium, and the bacterium starts proliferating there and produces a toxin which is a neurotoxin. It enters blood and it reaches central nervous system and it causes spasm, generalized spasm in the body. So that's how tetanus is clinically manifested. And another type of tetanus, sometimes tetanus is in the adult person, sometimes it's in the neonates after birth. And in this case, it's usually not due to wounds or puncture wounds or abrasions or things like that. It's due to unsanitary conditions that under which the delivery has been done, especially in the old times, contaminated scissors that uh, midwives have been using to cut the umbilical cord. So the Clostridium tetani produces a toxin, and the way that we fight this, the way that we try to prevent this from happening is by vaccination. And the vaccination that takes place in here is by using sort of a altered form of the toxin of the bacterium, which is called a toxoid. A toxoid is something which looks like a toxin. So it's a toxin which has been treated with some chemicals so that it's lost its toxicity, but it has retained its immunogenicity. So if you inject the person with that, the person will start producing, the immune system will start producing antibodies against that toxoid, which can also bind to the toxin. And then if the person gets exposed to the Clostridium tetani bacterium and the toxins, those antibodies will able to inactivate bacterial toxin. It's called neutralization, so making it neutral. It means that those antibodies will be able to bind to the toxin and prevent it from reaching its target, which is central nervous system in this case. By the way, the antibodies which act against toxins are called antitoxins too. So that's one of the major ways that antibodies work, and it's also the case for some other bacteria which produce toxins. This is a picture from CDC website again, from a kid with diphtheria. So diphtheria is caused by Corynebacterium diphtheria. So it's a bacterium which sort of causes infections in the throat. It generates a sort of a leather-like membranous structure in here, which is basically a collection of bacteria which are proliferating. So it releases a toxin similar to the Clostridium tetani, and that toxin enters blood, and it can influence internal organs. And unlike Clostridium tetani's toxin, which is a neurotoxin, Corynebacterium diphtheria's toxin is a cardiotoxin, so it influences heart. And again, it has a high fatality rate. Again, for prevention of diphtheria, vaccination is the solution. And similar to tetanus and the tetanus vaccine for Corynebacterium diphtheria and its toxin, we use toxoids. So again, chemically altered toxins of Corynebacterium are injected to the person, and then antibodies are produced. Those antibodies have the ability to neutralize the toxin. So that's one of the major ways that antibodies work. Can neutralization happen for the entire organism. Like in these two cases that I mentioned, antibodies are binding to toxins, which are small molecules. Can antibodies neutralize the entire organism, the entire microorganism? Yes, if it's very small, and that means viruses. So antibodies are able to neutralize microorganisms, but not big bacteria or fungi or worms. They can neutralize viruses and prevent viruses from binding to their target. So neutralization by antibodies is one of the ways that antibodies work. And it can happen for bacterial toxins or for viruses. For instance, if you inject somebody with hepatitis B vaccine, antibodies will be produced against hepatitis B antigens, which can inactivate hepatitis B virus after it enters the body. They can prevent it from binding to their target cells, which are liver cells in the case of hepatitis B virus. So that's one of the ways that antibodies work. Another picture that I've got from WHO website shows another important infectious disease, poliomyelitis. Again, a viral infection. It's a fecal oral infection, so it enters body through gastrointestinal tract, and it causes basically the death of neurons in the anterior horn of the spinal cord, and it leads to paralysis. And these kids that you're seeing in here are cases that have had polio for years. So in the beginning, it's only muscle weakness and sort of flaccid paralysis, but after some time it can lead to deformations. And you probably know that polio has been a major health issue in many developing countries for decades. Fortunately, it's almost eradicated in most countries, but in the past it's been a very, very major health issue. There are two types of vaccines for polio. One uses the dead virus, and it's called IPV, for inactivated polio vaccine, and it's injected to the person. So some people call it injectable polio vaccine. So it's dead virus. So if you inject somebody with IPV, antibodies will be produced in the blood that will neutralize the virus after it enters the blood. So the virus enters through contaminated water or food. It enters the gastrointestinal tract, and then it goes through the intestinal epithelium and it enters blood. And after it enters blood, it will circulate until it reaches the target cell, which in this case are neurons of the central nervous system. So if somebody's received IPV, antibodies will be present in the blood, and they will inactivate the virus in the blood, similar to what we mentioned for inactivation of bacterial toxins, for example. But another type of vaccine is the so-called OPV, or the oral polio vaccine. And this is the vaccine which uses an attenuated, weakened virus. And in contrast to IPV, which is injected, OPV is given through basically oral routes. So the virus is sort of attenuated, it's sort of weakened, it doesn't cause any disease, but it stimulates immune system. And by stimulating immune system at the surface of mucosa, it generates type of antibodies which can neutralize or inactivate the virus in mucosal secretions. So it doesn't even allow the virus to enter blood. It inactivates the virus at the surface of mucosal secretions. So that is what OPV does. So we have two neutralization mechanisms. One happens inside body in the sort of in internal body fluids, like in blood or plasma. And another type of neutralization happens at the surface of mucosa, because the antibody which mediates this type of neutralization is secreted into mucosa. So neutralization is one of the mechanisms, major mechanisms that antibodies work. But is it the only mechanism? No. A lot of pathogens do not respond to neutralization. And here I have a picture. It's a chest x-ray from a patient with pneumonia, bacterial pneumonia. So you see this opacity in here in the lung. It's a patient who has been admitted to emergency department with high fever and difficulty in breathing and cough with sputum and chest pain. And the x-ray is showing this. And when you look at the sputum of that patient, you see a lot of bacteria which look like diplococci in here. And this is a case of bacterial pneumonia caused by Streptococcus pneumonia, which has been traditionally called the pneumococci. And pneumococci do not only cause a lung infection, they can also cause meningitis, so it's a very important pathogen. So what happens for pneumococci when they enter the body? Well, antibodies bind to them, but antibodies by themselves cannot neutralize them. If the bacterium is too big, it's not like a toxin or a tiny virus that the antibody can bind to it and sort of covers its surface and not allow it to do anything. So antibodies by neutralization cannot do anything. Pneumococci should be phagocytosed, and here I have sort of a schematic. Imagine that it's the pneumococcus or bacterium. They can be phagocytosed, but their phagocytosis is not very efficient. Here, antibodies collaborate with phagocytes, so they cover the surface of the pathogen, and then the phagocytes have receptors which are called Fc receptors. Fc means the constant fragment of the antibody, which can bind to the tail of these antibodies and internalize the microbe. So by binding of antibodies to the target, they can enhance phagocytosis by hundreds of times. So they make phagocytosis much more efficient and potent. And this is the process that you've heard about. It's called opsonization or opsonization, and it's one of the mechanisms that antibodies work. So if you cannot neutralize it, you can make it easier for phagocytes to target and eat the pathogen. So that's another mechanism by which antibodies work. In addition to neutralization, you can opsonize pathogens and facilitate their phagocytosis. Do we have any other mechanism? Yes. This is a picture of a child which has been admitted to emergency department with fever, headache, vomiting, and reduced consciousness, and a lot of subcutaneous bleeding. And this is an example of the so-called meningococcal meningitis. It's a type of meningitis which happens after infection with a particular type of bacterium, and it's a very aggressive and fatal type of meningitis. It's caused by so-called Neisseria meningitidis. And Neisseria cannot be neutralized. They can be opsonized. Their opsonization is good, but it's not sufficient. So what do you do with the bacterium if you cannot neutralize it? And if the opsonization and phagocytosis is not sufficient? Well, one way is to punch holes at its surface. So the antibodies can call on some particular type of proteins which are called complement proteins. You've had the class about complement proteins. And the complement proteins can sort of aggregate at the surface of the cell and sort of generate holes at the surface of the bacterium, and it will kill the bacterium. And they also produce other things like tactic molecules and some other molecules which also help in the process of defense against the bacteria. So if you cannot neutralize it or the opsonization is not sufficient, you can try to punch holes at the surface of it by activating the complement. Another mechanism is so-called antibody-dependent cytotoxicity. Well, here I have a picture from a patient which has hepatitis, and you see the jaundice and sort of the yellowish discoloration of the sclera and the skin here. Viruses usually enter cells and live inside cells, and as we will see in the immunology of infectious disease class, when something is inside the cell, the antibodies cannot have access to. When they are outside the cells, antibodies can neutralize it. But if once they are inside, they cannot do much about it. And then it's the responsibility of other types of immune cells, including T cells, to get rid of the infected cell. However, when this virus is coming out of the infected cell, and it's called the budding process, budding, budding process, for a limited period of time, the antigens of the virus are exposed to the antibodies, and antibodies can go and bind to them. Once the antibodies are bound to the surface of a cell which is showing viral antigens, two things can happen. One is that complement can get activated, and by punching holes at the surface of the infected cell can kill the cell, and also viruses inside it. And another thing is that we have a couple of cells which like the antibodies which are bound to surfaces. And one of these cells are the neutrophils, the same ones that phagocytose, and other ones are natural killer cells. They come and bind to the tails of the antibodies, and they release the toxic granules inside them, and in this way they kill the infected cell. This is called antibody-dependent cytotoxicity, a type of cell killing which is dependent on the presence of antibodies. So this is another mechanism by which antibodies can exert their function. So binding to the surface of infected cells and killing them with the help of other leukocytes. And finally, I have a picture from a boy here which has had chronic fatigue, anemia, pain in the abdomen, a big liver and a big spleen, and also a high number of eosinophils in blood. This is an example of an infection with a worm. And the worm that I have in here, it's a particular type of worm that infects liver. We have worms as one of the major types of pathogens. Well, non-microbial pathogens. These guys are too big. They are not microbes, but they're still infectious agents. So worms are one of the major groups of infectious agents. How can we deal with worms? Well, worms cannot be neutralized. They are too big to be neutralized. You cannot phagocytose them, so opsonization is not going to work. You cannot activate complement at their surface. Well, you can activate complement at their surface, but you cannot punch holes because these guys have a very thick skin. So punching holes is not going to work. So what can we do with them? You cover their surface with antibodies. Well, a particular type of antibody. We will see it later. And you might already know that it's IgE. And we have a couple of cells which usually they have the receptors for the tails of this particular class of antibodies, eosinophils and mast cells, and they bind to the surface of the worm. Well, in reality, these are very tiny things. You have hundreds or maybe thousands of them covering the body of the worm, so this picture is not drawn to scale. And they release their granules, and these granules can kill the worm. So this is the defense against worms. And in a sense, it is type of an ADCC. It's a specialized form of an ADCC. What is a specialized form which works against worms? So that's another mechanism by which antibodies work. So we have multiple different mechanisms: neutralization, opsonization, complement activation, and ADCC, as well as degranulating, emptying the granules of eosinophils and mast cells. And different classes of antibodies that we have talked about are related to these different types of functions. So each of these classes does one or some of these functions better. IgA is the major antibody which is present in mucosal secretions, and its function is to neutralize pathogens in the secretions. IgE is the one that protects us against worms. And of course, if you have too much of it, there might be a possibility for allergic disorders. IgM is very good in activating complement. Well, these sort of arrows that I've drawn in here, so the thickness of the arrow is kind of, I'm trying to show the potency or the efficiency of that function. So IgM is very good in activating complements, but other things, not very much. Can neutralize, but not very well. And then we have the major antibody, which is quite multifunctional. It's the best antibody for neutralization, IgG. It's the best antibody for opsonization, for complement activation, and also for ADCC. And also one major function of IgG, as you know, is during fetal period. It can traverse placenta, and from maternal blood, it can enter fetal blood, and can have a major effect in protecting the fetus and the neonate after birth against pathogens for quite a few weeks. So these are different functions of the antibodies. All right, so this is the end of B-cell story. It was a bit of a long story, but as I mentioned, one reason that this class was a bit long was that we had combined the B-cell and T-cell development story in here. So yeah, well, this is all what we have in here. And in the next class, we'll be talking about T cells, development, activation, and function. Well, the development of T cell, I don't have much to tell you about because it's very similar to B cell development. So we will mostly focus on T cell activation and T cell function. In summary, What we learned this class was that we have two major mechanisms that give rise to billions of different forms of antibodies at the surface of B cells. Those are combinatorial diversity and junctional diversity. After B cells leave bone marrow, they go to secondary lymphoid organs, and there are some differences. B1 cells usually go to peritoneal cavity and mucosal surfaces, but conventional B cells go to spleen and lymph node. After B cells see their antigens, they proliferate, they differentiate and produce antibody, and it will depend on the nature of the antigen and the involvement of T cells. The type of B cell response that we'll be having could be T independent or T dependent. And then finally, different classes of antibodies use different mechanisms to protect us. Sometimes they neutralize, sometimes they opsonize, sometimes they activate complement, and sometimes they do ADCC. And one type of ADCC is against worms, which entails degranulation of eosinophils. So that was the end of the story. These are the references that we use for this course, and I hope you've learned something from this course, this class. Don't worry too much about the questions in the exam. The questions will be kind of general conceptual questions, and I guess you should not have much difficulty for answering those questions. Okay, so till next time, goodbye.